Single-cell microaspiration is a method to complement the different techniques used in the field of microbiology in general and in virology in particular. It could answer key questions and resolve problems encountered during giant viruses isolation by separating a virus from a viral mixture, presenting a low abundance virus as it was the case for two novel viruses, clandestinovirus and usiopathy virus, from another one, which is faustovirus, present in high abundance in a whale. The main advantage of this approach is the use of an indirect strategy. It consists of separating and cloning the infected hosts to obtain a viral separation. So, this method monitors the different steps from capture to the release of cells and confirms the sorting process by microscopic observation. Finally, molecular biology is used to confirm the sorting and electronic microscopy to observe the separated viral particles. This approach resolved the major problem of virus separation. It is a perspective for cloning amoeba or cloning protists in general. The person who is trying to use this technique for the first time must be rigorous and precise when controlling pressure. The visual control is essential to confirm the capture and the release of only one cell.
Use Vermamoeba vermiformis strain CDC19 as a cell support. Add 30 milliliters of PYG medium and 3 milliliters of the amoebae at a concentration of 1 × 10⁶ cells per milliliter in a 75 square centimeter cell culture flask. Maintain